Thank you all for joining today. So today I'd like to tell you about a major research project that we have done, and I'm the lead, but by no means the only one that deserves credit. We have a very strong team of people here, researchers, Dr. Jonathan Koch, Dr. Lindsay McCabe, Byron Love, Dr. Kelsey Graham, who some of you may have met when she did her postdoc there in Michigan at MSU, and Craig Hunsinger. And we were fortunate to be funded on this project by Project APIS-M and Costco. And I'll follow up with that more in a little bit. We also have a very strong collaborator, a commercial beekeeper, Darren Cox, of Cox Honey LLC, Utah. He's located up here in northern Utah near us. Besides this team, we had a large number of great workers on this, so I need to reach out and thank everyone for all their great help. Those people in bold in particular took a lead role out in the field or helping us in the lab as well. So without these people and without the funding by Project APIS-M and Costco, it would never have happened. And as Anna said, our group here in Logan, we are the home of the U.S. National Pollinating Insect Collection, i.e. the bee collection for the United States and North America. And as many of you know, there's tremendous diversity in bees, over 20 different thousand species worldwide. In North American, we have over 4,000. We're still discovering new species that we didn't know exist. And each of those is very important for us, for both as keystones or pollinators in our agricultural systems, but also in our natural ecosystems. We depend upon their pollination activities to keep, provide food for us, but also to keep the plants that we depend on in our environments. And as you can see, I'm showing honeybees here as one of those essential keystones. It is our major pollinator of commercial ag crops in the United States. And as some of you may know, this diversity of bees is not uniform across North America. What I'm showing you here are gradients in number of bee species. The darker the colors, the purples, the blues, means that there are many, many more bee species. And it goes against what you might think, that a lot of these bees are found in the American Southwest, which is where a lot of the deserts, extreme environments are found. We just see a great diversity there. That doesn't mean that places like Michigan and further east are lacking in that, because we are finding new species back there as well. So in terms of bees, as Anna mentioned, I was one of those people that got involved early on with colony collapse disorder, which was around 2006. In almost that 20-year time here, we've made tremendous progress in understanding what might be underlying decreased colony survivorship for honeybees, but also decreases in other bee species and their populations and their status. As you know, in Michigan, you are the home of the rusty patch bumblebee. It and others have been added to the endangered species list. And we have a lot of concern about bees and keeping them in our environments for this pollination. We'd like to attribute the stressors that impact the bees to these four main Ps: pesticides, pathogens, parasites, poor nutrition. And we know that they interact with each other, that can compound and make things difficult to separate out. Climate and climate change, with the dark blue rectangle here, square, does overlie all this and is having a greater impact. We now think that poor nutrition, the lack of floral resources, and climate are one of the major threats to bees and impact him a great deal. And it comes down to that we need a lot more bee pasture, floral resources to sustain not only honeybees, but the other bee species as well. And this is not just in the US, but it also exists in Europe. And come back and talk about that a little bit here. So in terms of the bees here, so everyone's on the same page, we have three major groups of bees. Honeybees, which many of you are very well familiar with, is basically a social animal. It lives in a colony. It takes the entire colony to exist. So you have the one reproductive, the queen bee who lays the eggs, the larvae hatch are taken care of by workers. They develop, the females go out and take on new tasks as they're adults, and the males are mainly reproductive. And they go out and forage for pollen and nectar. That nectar is what they make into the honey that we all enjoy. Honeybees were brought to the United States in over 500 years ago by the Spaniards and the Jesuit priests. They brought them with them when they came over to North America, primarily for the wax for the candles in the church. So they have been in North America for over 500 years. Even before then, in the Miocene, we do have a fossil coming out of Nevada that is a member of the Apis genus. So Apis has been in North America for a long time. Bumblebees are also social, but they're an annual colony. The queens emerge from diapause in early spring, and she at that time point is like the single mom. She has to go out and collect nectar and pollen in order to provision nest cells, lay eggs, rear up the workers. When they emerge, they take over the task of collecting pollen and nectar, coming back, and she becomes sole reproductive. Late in the summer, fall, she switches over to producing the future reproductives, the queens or the gines, and the drone bees. And they mate before winter occurs, and then those future gines, queens, go into hibernation. The majority of our 4,000 bee species are solitary bees. They either live in cavities above ground or create nest cells below ground, in the ground. And in this case, each bee, each female, is a single mom. She goes out and finds a nesting site, collects nectar and pollen and brings it back, lays an egg, seals off that cell, goes on to build additional cells. She never interacts with the offspring in the same way that bumblebees, honeybees do. These bees, all the different species, are not active at the exact same time and period. Some of them emerge very early spring, other mid summer, some towards fall. And you just have incredible diversity in how they exist. And for both bumblebees and solitary bees, it's very difficult to find those nests out in the wildlands. The commonality here between the solitary bees, bumblebees, and honeybees is that they all need pollen and nectar for reproduction and for their survival. Recently, in the last 5 or so years, there's been a lot of debate about how honeybees impact other bee species. And there's been various publications in the literature, research done to suggest that there is a negative interaction competition. Well, some of this literature has come out of Europe, where the honeybee, Apis mellifera, is a native species. So I'd like to put forward that this is not necessarily a bad bee and impacts on the other bees, but more so a question of how many floral resources do you have? How much food is there available there, that carrying capacity? The way of thinking about that, if we were all at one meeting, and I think there's over 100 people on the webinar here, and we wanted to have lunch and only 10 sandwiches were delivered, we would have a great exceeding of our carrying capacity. We'd be competing for those 10 sandwiches and just exceeding how many mouths are fed. So in this diagram here, this illustration, what I'm showing is a platter with flowers on it. And you can think that there's a set number of flowers out there at any point in time that endemic non-managed bees and managed honeybees might be wanting to make use to get that pollen and nectar that they need for reproduction. And what determines how many flowers there are a lot of different factors: weather condition, temperature, and moisture. There may be also other organisms there that want to eat those flowers that are also competing for it. So herbivores out there, large mammals, cows and sheep, which I'll talk about here in a little bit, or use of the land. There may be a recreational area in there that a lot of dust being generated that may impact those plants. Amongst the bees overall, we do have specific plants and their bees where they have a very intimate relationship. They're specialists. Example of that here in the West is the death camas lily, which has only one bee species that can pollinate it. It produces an extremely toxic nectar and pollen, and that bee has developed a way to be resistant to those toxins. Honeybees cannot pollinate it. So one can think about also this need to keep those specialist bees that pollinate specific plants out there around in order to keep the reproduction of those plants. So what I'm going to put forward here is trying to define or understand more carrying capacity and what the interactions are. So Project APIS-M approached us to ask if we would do this research. And I worked with my national program leaders to get their approval of this. I also sought outside stakeholders from all different avenues, both the conservation groups and honeybee keepers and others, to provide input on the project to make sure that we did it in a very balanced way. We agreed to take on the funding with the stipulation that we were going to do the best quality research that we could in an unbiased fashion, so not taking any side up front, and that we would have the freedom to publish any results that we found, no matter any way that they went. So I'd like to show you what we found here. We asked three main questions, or three areas here, about how the honeybee's apiary can impact native bees. We asked about reproduction and colony growth, not only for the native bees, but also for honeybees. How did them interacting with each other impact one another? We also asked how they interact with the plants. Do we see a change in how they're foraging or pollinating the different plant species? And lastly, we asked about disease and pathogen spillover. Most importantly, what I'm going to show you today is we ask about impact of those pathogens in the different species. And that, I think, is brand new data that I'm quite proud of. So get your feedback on that. So lead it off. How did we do this? We did it two ways. We did it in cages. And the reason we did it that is that we wanted to generate competition. We wanted to see what it looks like in terms of how it impacted the different species, and look at also what the impact of the influence of flower species were. And the reason why our lab is really good about doing this research is that we not only know how to work with honeybees, but we can generate bumblebee colonies and also able to create larger populations of specific osmia or solitary bees by themselves and monitor the nesting of and reproduction of all these bee species. We also set up studies out in the field, and we did this out in U.S. Forest Service land and private ranches. We had 3 sets of apiary sites. The first year we just did it at one of the sites because this was a very complicated experiment and collection of data. We wanted to make sure we got it right. And then we expanded it out to 2 additional sets of sites, years 2 and 3. We also, by doing this, we're able to look at, we were, I guess, experienced different climatic conditions in those 3 years with extreme drought, severe drought, and moderate drought. And that was important. So who did we use? So these are the players, the bees that I'll be talking about. We have Osmia bruneri. Unfortunately, there's not a nice common name. It is a solitary bee. We can create these nesting blocks here, these wood blocks that are drilled a certain hole size, and we can stick little straws in, and these bees will nest in there. It's a really pretty little bee. It's a metallic green-blue, as you can see here. It uses masticated plant material to create the partitions in its nest. And so we could monitor the nesting here, and we could remove those nests by pulling out the straw when they're full and replace it. So there was a continuous supply of nesting sites. The other bee that we also worked on was Bombus huntii. It's called the hunt bumblebee. And it is a native bee here in the West. It's also has been brought into commercial production and is available in Canada with BioBest. It's a great pollinator of crops. We were able to take field caught queens and rear them up, get them to establish and form colonies. So part of my group here, I like to call them the bee whisperers, are able to generate the right conditions to have this happen in. These can get very large over the years. So showing in the middle picture here down in the bottom left, that a large colony. We figured out a way to put these colonies out in the wild and protect them. And I'm showing you there, we put the cages in to these Rubbermaid containers. The bees could exit and go out. We could also monitor them using this borescope here. And that was important because bumblebees are actually more defensive, I would claim, than honeybees. They can sting multiple times and they don't forget. So being able to go in and peer in and see what the colony was doing, how it was growing with the borescope was ideal to monitor it. We also stuck a sheet underneath the cage down in the colony to collect the fecal material or frass. And that was important to our success looking at pathogens. On the honeybees, our collaborator, Darren Cox, established apiaries out in the forest of 48 colonies each. In our cages, we established full-size colonies in these modified Langstroth boxes here and equilibrated each of those colonies to having the same number of adults, equal number of adults going in when we began the cage studies. And we were able to work with them just like you would do in other research. So in our cage studies, we set them up the first year over just Phacelia, shown here. In the other years, we found that we needed to expand that out to include all these species here. These were very large cages that we could put out and irrigate and monitor the bees inside these cages. And this gives you an example of what we saw there and how we set up both the solitary bees, the honeybees, and the containers containing the bombus there. In the field, this is a map, an aerial view off of Google Maps here, Google Earth, showing our 3 apiary sites. This is, these were located to the east of Logan, so up into the mountains. The large blue area there is called Bear Lake. It's extremely large natural lake. It's become a hotspot for recreation. We're about an hour and a half away from Salt Lake, so a lot of people come up there, vacation homes, boating, etc. But a lot of people also visiting the national forest. So on these 3 apiary sites, Blacksmith Fork, which are the pink dots there, are important. The Blacksmith Fork apiary is on a private ranch and it has existed for over 20 years, that apiary. And in any one season, outside of our research time, it would have at least 96 colonies, sometimes twice as many as that. And that's important. The Blacksmith Fork non-apiary site is on Forest Service land and has no history of honeybees being placed there. So for each of these sites, we have a honeybee apiary, shown here for Twin Creeks and Twin Creeks non-apiary. It's located right off the highway that leads over to Bear Lake, a lot of traffic going through recreational. And then we have to the north, Franklin Basin Apiary. The apiary is off of a Forest Service land. The Franklin Basin non-apiary is just beyond a private ranch, also on Forest Service, but it's right like 1 mile outside of the Idaho border there to give you an idea where we're located. Within each of these 3 sets of sites, the way we set them up was like this bullseye pattern. In the direct center here is where we put our honeybee apiary and bumblebees there. So we had sentinel bumblebee colonies located with the honeybees. Out at 5 kilometers away is where we had a second apiary with just bumblebees alone. In between and each of those apiary sites is where we positioned our solitary bees and nesting boxes. And those were seeded with females and males in equal numbers. And what this allowed us to do was to monitor the impact of the honeybees and ask about distance, how that related to the bee success as they got further and further away from the apiary. We also asked about the endemic bees that lived in the area, who they were and how many. And this was important because we really had never done a full-scale survey of native bees in this particular area. So this is providing some important information back just on what bees are found in the forests out in this area. We have over 9,000 specimens that we have now collected, pinned, and have identified. We went out every 2 weeks, put out bolt traps. That's what these little colored solo cups are. They're filled with a soapy water solution, attracts bees and other pollinators that fall in there. And we also, in a very defined grid area that we use throughout the seasons, went out and netted bees off flowers. We also did identification of the flowers and floral counts in those same grid areas over the season. So we're, by those floral counts, figuring out exactly how many floral resources were around, how much food there was for the bees in that area. Okay. So I grew up in the West, and I thought I knew everything there was, but of course I don't, and that's obvious. But I didn't fully understand how many uses there are for the U.S. Forest Service lands. So I'm showing you here their mission statement, but there's also this slogan, land of many uses. I can attest to that. This is very true. So I'm showing you a GIF here. It's just repeating over and over. One thing when we're doing this research, it was during COVID, and a lot of people had cabin fever and wanted to get out, and it still exists. So on these Forest Service roads, which sometimes are questionable and how easy it is to get across, these people would come out with their RV vehicles, their tents, etc., and find any place to camp off of this, not in a developed campground. A lot of these people would come out with groups or friends and bring their ATVs with them. So what I'm showing you here is the aftermath of one of those clubs of ATV people driving up the road, and you're seeing the dust clouds come out over the floral resources here. We know from published research, there's 2 papers out of the United States and one out of the United Kingdom that shows that dust and other pollutants coming out over the flowers does impact the quality of the pollen and productivity of those flowers. So that's one of the things that we found we had to contend with. The other thing was that all these areas, the private ranches and the Forest Service, had active grazing permits or cattle and sheep placed on them. Excuse me. And there were the apiary permits. Jaron Cox had his historical use and was permitted to do this. And there are a lot of other uses too. Excuse me. So to give you a little bit of background, my family, they were pioneers in Colorado, and my grandfather ran sheep up in the mountains of Colorado. And we had him a little bit as a kid. What I didn't understand was the impact potentially of both sheep and cows on floral resources. At our non-honeybee apiary site up in Franklin Basin, they brought in 7 truckloads of sheep while we were doing our research and offloaded them and then herded them up the valley. I'm showing you the before and after. The sheep came through and ate every flower off of this horse mint that it's blooming here. Unfortunately, it did not bloom again. The cows, especially in extreme drought, were looking for anything to eat. So all that trampled plants there, that's not my crew. The cows were just looking for forage. So there is this interaction, competition for those floral resources by other things besides honeybees. So with all of that, what did we find? Our first year, we put out our colonies, our bumblebees and the honeybees and the solitary bees. Within 2 weeks, we had a major surprise. We found at both the honeybee apiary site and the non-honeybee apiary site, this bee showed up. It's a cuckoo bumblebee. It's a kleptoparasitic bee, and I would claim that it's actually an actual predator. It moved into our Bombus huntii colonies. The citrus bee here lacks the structure needed to gather pollen nectar. The only way it can reproduce is to find another colony of a different species, come in and kill the queen, and take over the colony and have it rear up its offspring. And that's what happened with all of our bumblebees, most of our bumblebees at both the honeybee site and the non-honeybee site. We made lemonade out of lemons here. So we took those queens that invaded and we actually did population genetics and counted out how many we found. And what we discovered was that there was equal genetic diversity at both the honeybee site and the non-honeybee site. And why that is important, citrus here is an apex predator. So if the honeybees had had an impact on the bumblebees that citrus depends on, the other species, it should have shown up in the genetic diversity, that it would have been reduced at the honeybee apiary, and that was not. So this was very strong evidence suggests that honeybees apiary, this 20-year apiary that had been there, had not impacted in the previous season or had a long-lasting impact on the bumblebee populations there. So that first year was important because we came back and figured out how to make an excluder to keep citrus out of our colonies. That's what this little yellow slider is. It has a hole in it that the huntii workers could go in and out, but keeps the citrus out. We were able to use that and move our research forward. We had enough backup colonies to replace the ones that we lost that first year. So that was important. That's what I'm showing you. There's a little pollen trap here and a slider that we can control who gets to go in and out. So with that, what did we learn? In our cage studies where we generated competition, we saw that all three species, the honeybees, the bumblebees, solitary bees, were impacted. There was decreased reproduction of the osmia and decreased colony growth for both the bumblebees and honeybees. We didn't see a winner come out of that. Everybody was a loser when we exceeded that floral capacity, carrying capacity in those cages. In the field studies, as you can anticipate, as you guys know who are beekeepers, there was a loss of colonies, primarily due to queen loss, that they died out, about 10%. The bumblebee colony and its reproduction were not impacted by honeybee apiary. We did have a higher than anticipated failure or death of colonies at all the sites. And I'll come back and tell you how that relates back to honeybees. We did not see an impact of the honeybee apiary on the reproduction of our solitary bees. They did equally well at all, both the apiary and non-apiary sites and the sites in between. When we also did not see an impact on other bee species and wasp species that were using our nesting blocks as well. So what this says, if we do exceed carrying capacity, bring in too many bees, yes, indeed, you can overload the environment and have impacts on reproduction of all the bee species. But with the 48 colonies in the apiary, that we did not see an impact on native species in forest lands, even under extreme drought. The climate and the drought temperature did have a significant impact, though. So as I said, we did have drought. It's very dry in the West, and 2020 was moderate drought. 2020 was extreme drought. Very low snowpack, very low rain moisture. One of the driest years on record. 2022 was severe drought. One thing that we did notice was that the honeybee colonies were being brought into the forest earlier every year over across these 3 years. Things started much sooner just because of the shift in the snowpack and seasonality. We did find that the drought impacted the amount of flowers that were available there. 2021, there were significantly fewer flowers. And the honey yields in the extreme drought year were down 60%. And as I'll show you a little bit, the nesting of solitary bees was greatly impacted. Darren Cox agreed that I was okay to share this data here. The most important thing is there, just to show you down below, is that 2021, that honey production dropped way off. Our solitary bees, we counted how many nest cells were produced at each of the sites. And with moderate drought, you can, on the graph on the left, you can see it's much higher. 2021, significantly lower compared to all 3 years. But the biggest surprise to us was what happened when we looked at number of females versus males. So solitary bees are just like honeybees. That female, she can lay a fertilized egg that develop into a female on down the road. She can also lay an unfertilized egg, develop into a male. And in the solitary bees, often the sex ratio is 1-to-1, one female nest to one male nest for that female. In the extreme drought year, it shifted dramatically to 1 female to 3 males. What that translates into is that that drought impacted the potential reproduction of that bee even more so. There were fewer progeny being made, but many of them became males, not females. So there were fewer females being made for the subsequent year. And these are ones where we seeded the nest block. We weren't looking at what was happening in the endemic bees. We'll still be analyzing that. So climate is a major factor here. So we also, the second part here, asked about changes in foraging behavior. Our cage study, we could actually go in and monitor it, sit there and watch where bees went. And we did that over several times. We did find that the solitary bee, Osmia bruneri, shifted for 2 plant species when there were honeybees and bumblebees in there. It increased visitation to 1 plant species, increased it for another. But we didn't see shifts in the honeybees or bumblebees and what they're visiting. We did ask, what were the interactions on flowers? Did we see a bully among the bee species? We know that there are other species like the Anthidium bee, which has been introduced, that it defends a patch and it keeps other bees from coming in to pollinate the flowers. We didn't observe that at all. All these 3 species, the honeybees, bumblebees, and the osmia, got along great on the flowers. The only negative interaction we saw was in flight, where because we had so many bees in there that we did observe mid-air collisions. I told my group we should have put in air control traffic for there, but that was my attempt at a joke here, so forgive me. In terms of how they did on the plants, reason why the first year we just used Phacilias, we knew that Osmia bruneri could do quite well. But to our surprise, honeybees and bumblebees, the colonies just declined over the year. They did survive when we just gave them that one plant. When we expanded out to all the different species in there, they did great. So this supports other research that other people have done, saying that you get greater immunity, greater reproduction, more healthy bees when you have a diversity in the host plants or the flowers that are out there for the bees to forage on. In the field studies, we've now done the identifications. We're still doing the analysis to ask what are the interactions. But just to show you, we do see some preferences and differences in there. So here I'm showing you for one of the field sites, what were the visitors to horse mint, a rabbit brush thistle, and potentialia, and other plants that were blooming in the area. You can see that the native bees did differ a little bit. Horse mint, everybody liked. The rabbit brush, potentialia, and the mallow were also favorites. Thistle, however, was a little bit different. This is an invasive thistle. This was on the private ranch. And it had unfortunately had this non-native thistle introduced to it. And it turned out it was the non-honeybees that were mostly attracted to it. So there is going to be some differences in who's visiting what and how they're involved. So pathogen transfer. One of the big things that's been in literature is that, yes, honeybees do have a lot of different viruses and diseases, and we know that these can be shared. And so people, including myself, in the past have speculated that this could have negative impacts on other bee species. We wanted to address this in particular. So viruses are what we focused on. It's my main expertise. And there have been over 40 different viruses described in honeybees worldwide. Most of these are single-stranded RNA viruses, very teeny tiny ones that you have to use an electron microscope to see. And you can think of them being sort of hard to see, just like COVID or polio viruses. So most of these viruses have been discovered because there were isolated lost cases or losses of bees. And we don't really understand fully what their symptoms are. Some of them, like sac-brood virus here, that's what the drooping picture of the larvae is, have been known in the US since at least 1900s, early 1900s. Deformed wing virus became apparent when we had Varroa destructor introduced into the United States. So it takes the parasitization of the mite for the bees to develop these deformed wings. And we don't really understand what they do and how they impact honeybee colonies, but we know even less for other bees. There's also a DNA virus, and this is actually a very large virus. You can almost see it with a microscope or a light microscope. And it was found here in the United States in the 1960s in honeybees. So it was given the name Apis mellifera filamentous virus. And it's a very large DNA strand. That's what you're seeing on the picture of the spaghetti on the left here. That's the DNA inside the virus particles. So this was described in honeybees. It's also been found in other non-Apis species. We now have genomes for it. And nobody had been really paying much attention to it. And we began to look for it in our studies. All of these viruses in honeybees have multiple routes of transmission. Your queen can be infected, lay eggs with the virus in there, and it'll show up in the larvae, the pupae, and the workers. If the queen's non-infected, she can acquire the virus by mating. So we do have sexually transmitted viruses in bees. Comes in through the sperm, infecting the sperm, and in the seminal fluid. We can also have the workers being affected and secreting the virus in their glandular secretions, so showing up in honey, the royal jelly, or in the bee bread. Lastly, for honeybees, the varroa mite is a vector and can be infected with some of these viruses. You as a beekeeper may also play a role in this if you use your hive tool or take your dead out colony and give them the frames of the old colony with still has food or honey in it, you may actually be spreading the virus. In other bee species, we know that this can also occur for a lot of them, where that we've been looking at it. When I was at Penn State, we found that there was another route of infection, a fecal oral route. We found that the flowers and the pollen on the flowers became infected with these viruses. Unfortunately, bees do defecate on flowers. It's been shown that that is also a way that you can get other diseases like nosema, varomorpha. Bacterial diseases can also be deposited in the fecal material. So this is a way to get viruses to go from one colony to another colony just because of that infected, contaminated pollen. But we also found at Penn State that it was a route to move viruses to other species. And I'm showing you here the double-headed arrows. We had evidence that this was a way to move viruses back and forth, like from bumblebees to honeybees, vice versa. And this has also been suggested to hold true for the DNA virus. So what did we do? We took samples from the honeybee colonies at the beginning of the season and at the end of the season. And what I'm showing here are matched data for individual colonies at beginning and end of 3 different sites. The Franklin Basin site, we have completed our analyses. I just need to get the data analyzed now to fill in that missing bar there. But what you'll see here is that the number of viruses that we detected, we looked at RNA viruses, that it was very high. These bees came in from almond pollination, were placed in the forest lands. And what we see by the end of the season, that the number of viruses that we could detect had significantly decreased. Was not only in the number of viruses, but in their prevalence, how abundant they were, but also the titer, how much was individual bees, went way down. So we do think that the colonies have an advantage when they get placed in a non-agriculture environment. It increases their health and they're able to cure themselves or have immune responses to clear out many of these viruses. The DNA virus, this filamentous virus, is different. We found it in every colony at both the beginning and end of season, and we didn't see any change in titers. What I'm showing here is the amount of virus relative to the bee DNA in our DNA extracts. And the axis here on the left, you'll see it's a 1 in 2021, meaning that there's 1 viral copy of DNA to each copy of a bee cell in there. So a fairly high infection. This is way beyond what we see with RNA viruses. 2022, this is an exponential scale that I'm showing you. It's 100 copies of virus to every bee cell. And in some of our colonies, there was colonies that had over 1,000 copies of the virus per bee genome. We think that this is a concern, that potentially this virus may be having a negative impact and be linked to some of the most recent losses that people have been reporting. In 1960, when it was described from colonies that were suffering major issues and disease issues. And it's been linked and reported back in other countries in Europe as also being associated with dead or dying bees in colonies. For bumblebees, we've also looked at it. So the way that we did this was we had queens that were collected at the same source, from the same source where we collected our queens, as those that went out in our deployed colonies. We also have that frass, that fecal material that were on the acetate sheets that we placed under the colonies and collected every 2 weeks. And it worked. We could actually find viruses in that fecal material. So what we found there were these single-stranded RNA viruses, and we also found the DNA virus. And we were able to ask what was the impact on the health of the bumblebees. So here I'm showing you over on the left, queens that were caught at the same time. We do detect these RNA viruses. There's less than 2 different types of viruses in each queen. When we put those colonies out in the field, we see an increased detection. So on the right-hand side here, in that graph, I'm showing you colonies that lived for the entire season. At the top, in the center there, colonies that died very shortly within 2 weeks after being placed out into the field. And at the bottom, colonies where the queen was lost during their time in the field. And what was critical was we do see that often that we find more viruses when they're placed with honeybees, But even in the non-apiary situation, when the honeybees aren't around, we're still seeing an increase in viruses. In some cases, larger number of viruses in that non-honeybee apiary site than in where the honeybees are. So we think that there is spillover from other bee species, both honeybees and other bees that are out there. The critical thing here is if you look at the colonies that stayed alive, they were the ones that had the most virus. And when we look at the fecal material over time, some of them did clear those viruses, others it stayed high throughout the entire season. So it's not associated with mortality. The dead colonies there, which died quickly, had relatively few or none of the viruses being detected. The queenless colonies had viruses in there. When we look at reproduction, though, we see that on the graph on the left here in the live colonies, that the number of queen cells is not significantly different as compared to the total number of RNA viruses that we're detecting in the colonies, that they're still able to make queen cells or reproduce. Even colonies that go queenless with the detection of viruses, they're able to reproduce. Of course, a dead colony is a dead end. It has no reproduction. The DNA virus, though, is a different story. The dead colonies, this presence of this filamentous virus was highly correlated with that colony dying, 80 to 90% prevalence detection there. And the live colonies had the lowest level, queenless colonies, the moderate level. If we ask about the source of this virus, did it come from honeybees? We find it's more often prevalent in the bumblebees that were not near honeybees. So we can say that it's not a result of pathogen spillover. So there's a lot more to be learned about this virus. For our solitary bees, we're still analyzing that data, but we have another project in the lab that has relevance to this question. So my graduate student, Mary Kate Williams, who's finishing up, we've developed a method where we can in vitro rear or do actual experiments with the blue orchard bee, Osmia lignaria. So in these well plates, we can take pollen and mix in either pathogens or pesticides, and then monitor an egg from development throughout its entire life cycle through diapause to emergence of the adults the following year. We also have done surveys for Osmia lignaria, which is a native bee out here in Utah. And we've taken newly emerged bees, cut them directly out of their cocoons. We do not detect any RNA viruses in those bees, but we do find this filamentous virus in those bees that have never contacted the environment in their adult lives. This indicates that it's a natural infection for the blue orchard bee or the Osmia lignaria. Doing these experiments, Mary Kate was able to ask about what the RNA viruses did. So I'm showing you in the graph here, larvae where she added in viruses and others that the viruses were present in the pollen or a part of the background there. We also added in this experiment an agricultural chemical, the organosilicon spray adjuvants. These are added into tank mixes when pesticides are applied, and we applied in different dosages in the pollen provisions here. These RNA viruses in bees that had just pollen by itself and the virus, it was cleared out by metamorphosis. We didn't detect it in pupae, but in those that we gave the organosilicones, we see that this virus persists. This suggests that in a commercial situation with pesticide application and these organosilicones, that these viruses may build up and may be important for the bees. We need to follow up now on that. For the DNA virus, we did add it into pollen provisions. We did find it was a significant mortality factor, that it killed the larvae. And we found the dead bees, both in the wild and from the experiment here, had the AMFV present. Most of the lignaria cleared it out, but it did persist through in the pupae and the adults and some. We didn't find it increased for the organosilicones, but we need to come back now with other pesticides. So just to summarize what this all meant was that yes, when the bees go into a field, a non-ag area, the honeybees, we see a decrease in the prevalence and titer of these RNA viruses. So these bees are probably having a significant benefit by being placed in a non-ag area. The bumblebees, there's a likely spillover from honeybees or other bees, but we did not find any impact of that virus on those bees or on the production of new queens, the reproduction. On Osmia lignaria, we could not demonstrate any impact on the survivorship. The DNA virus is filamentous virus. It's not just a honeybee virus. We need to do a lot more research to understand how it infects or impacts all the different types of bees and bee species. We know that it's highly prevalent, at least in the colonies that we have studied. Other colonies that Michelle Flanken's been monitoring up in Montana, she has not found it detected in many of her colonies. So we need to understand more how it's associated with different operations and associated with losses. We also need to look at it in terms of being a factor for bumblebee colony death and the death of solitary bees and how it's associated with other stress factors like actual pesticide exposures. And that's where our next phase of research will be going. So what overall summary here? How do honeybees interact with other bee species in wildlands? We know that if you exceed your carrying capacity, if you bring in too many honeybee colonies and don't have enough floral resources, that yes, competition can occur, but all the bees are going to suffer the consequences. In the wildlands, when we had 48 colonies per apiary, it did not impact the reproduction of sentinel bees or solitary bees. We did not exceed the carrying capacity, even under the extreme drought conditions. That climate, the extreme drought, did have a major impact on the flowers, how many there were, the reproduction of solitary bees, and also on the honeybee colonies in terms of honey production and probably overall health. If they'd been left out there, they would have suffered by not having enough flowers. We did not see any changes in foraging behavior, no direct evidence for exclusion of non-apis bees by honeybees on flowers. We still need to follow up and asking what happened with our pollinator networks when honeybees are present versus not. For pathogen transfer, we have no evidence for negative impact on honeybees on the health of the other bees. And we've learned a lot more about pathogens that we need to pay attention to here. So to summarize it, coming back to the carrying capacity here, that plate of flowers here, we know that how many flowers out there depends upon weather and climate, down here in the bottom left. We also suspect that other factors like dust from vehicles or pollution may also be impacting the quality of the flowers and amount of pollen. We do have major consumers, so the endemic non-managed bees that are present there, and our managed bee apiary insect livestock, as well as herbivores, both the non-managed and the managed livestock. So we need to balance out how many flowers are present and how many colonies could be introduced or how many herbivores can be introduced. That needs to be taken into consideration. So with that, I'd like to thank you all for inviting me, and I'd be glad to answer any questions. And if you think of things after the webinar here, feel free to reach out to me. This is my email here. I'd be glad to answer questions and have discussions with you. So thanks.